welcome to the new episode of Coffee House Experience. With us, we have Elise today. Elise is a postdoctoral researcher at UC Berkeley, working in, broadly in molecular biology, but specifically in genetics and, like exact sub area, I guess we talk about it here. So welcome, Elise, to our show.
Thank you.
Right. So, yeah, I guess we can briefly start with your brief introduction and sort of your brief area, and just curious how you got into this area research and how excited you are. So maybe you can just get started. Yeah.
Yeah. So first I was like really interested in biology, but I also really liked mathematics. So I did my undergrad in both.
Oh,
And
yeah.
then I
So
was
it's a major minor system. So you did your undergrad in France, I guess, right?
yeah. So it
Okay.
was kind of like a major minor. So my major was biology and my minor math. And
Interesting.
I really like evolution and population genetics. It's really a part of bio that has a lot of like probabilistic models that are used. So you can really describe biology and like what the genome look like with just some probabilistic models. So it's not a good description, of course, but it's just like an idea. So you'll have like mutations that happens randomly at different places and that have effects. So that will depend on different mechanisms that you can describe sometimes with mathematical equations. And so I did my PhD really in theoretical population biology. So I was like describing, using those models to describe how like a decline in a population. So when the population gets smaller, how does it affect the genomes?
Interesting.
But then I don't know, I really want to have like more big question to be more like data focused. And then that's how I end up in Berkeley with Priya Morjani. So she had like this huge data set of Indian individual, and were like so many questions we can answer. And also it was really nice to see like the impact, like people are really interested in what we are saying. It was a bit more than when I was doing my mathematical models. That was
Very,
really
very
nice.
interesting. Yeah, so very interesting because like typically like both myself and Srinjoy, we happen to study in the same university. I mean, we both of us are into engineering, and then I went to more computer science and he went to more like finance. But I mean, it's kind of a very interesting combination that you studied. Like, I mean, this is like I think very uncommon in our region that people would study both biology and mathematics at the same time. So it's like very, very interesting to know. I know that in population genetics and genome decoding, there are like a lot of combinatorial mathematics that you need to use and probabilistic modeling. So, it's kind of a very good to know that, people who have this kind of common interest, they can study this kind of very interesting subjects. So by population, when you say like there is a sort of, if a population is declining, by population sort of, do you mean any particular form of like, do you have a particular technical definition, or can you explain to us exactly?
So often in population genetics, what we call a population is just a group of individuals that reproduce together. So for my PhD, I was like, my idea was to apply it to conservation genetics. So just have an idea of like the population of these species that live in that place, are they in decline or not? So that was like the main focus. But it's true that it's quite a complicated definition. Like I think even in between biologists, we don't define it the same way.
Okay.
It's kind of like the species thing. We don't really know what a species is. If you,
Hmm.
we know for like easy things, but then
I mean, there
it's
is a
always a
gray area.
spectrum.
So there is a gray area, sort of, there is a not a common consensus, right? I see. Like by population, you mean like, for example, do you mean like a particular species, like division between species, or for example, like in human population, like for example, what could be a good example of population? Like Indian population is not one population that, if I, as far as I'm concerned.
Yeah, so that's why it's always a bit complicated, because you can then maybe call it a meta population because it's divided between a lot of group. I think we use population just to try to define a group. because we want to use models, you need to define a group to apply your model to. So that's a bit what is complicated. And all the population are different. Even so there were a lot of studies first in European, and if you just take like Icelandic people that are like on an island and not that many, compared to more like mainland Europe, it's also really, really different concepts. And like people are more different in the same population and things like that. So that's one of the things that is not well defined in our field.
Probably the Han Chinese are one example where you have a large number of people which can be defined as one population. As in, if you were to see their genomes and analyze them, do you define population in terms of a certain threshold of similarity in genome structure or something like that?
I think it depends on what you want to look at. So I think if you want to compare Asia and Europe, you can call Han Chinese a population, because it's like, because there will be a big difference. But I'm sure that in the different regions, like you can subdivide it for sure. I think that like what we did in India, so we had like first maybe the big state, like the region, but we can have divided by state, we can have divided by language. It's just that at some point you need to put a cutoff, and yeah, it's not always the same depending on what is your question. Right?
Yeah, so coming to the most crucial point of discussion, I guess, like one of your recent results, you showed you studied some modern population like data, right? So Indian genomes, like which are from modern Indians, not like the ancient studies, right? And I mean, I just read this article in the news outlet, online outlet called The Where, and they mentioned that your result shows that it's probably like a more establishment of some fact or some new fact that I'm not entirely sure of. So you'll explain that. So, and it says like just to give my idea of what I understood, that it says that Indian population is mostly they how do I say, like it's consists of three different ancient populations. So that's sort of the major constituents of Indian genome. So if you can like elaborate on that.
Yeah. So one of the things we do in population genetics, it's we try, like we call it like chromosome painting. We try to like color population by what ancestry they come from. And so in India, for the large majority of our samples, that is kind of like from north to south, we have kind of different ancestry in east and in northeast. But for that, like we call it like the Indian cline, it's mostly three ancestral populations. So one that will be related to some Eurasian steppe pastoralist ancestry, one that is related to more like Iran farmer ancestry, and one that is more related to South Asian hunter-gatherers. So that, yeah.
So, I mean, can you just explain a little bit more all these three populations, like when they, like what's their history or what you can talk about?
So how we do that is that we compare it to like ancient DNA that was found in archaeological sites. And we just like see, how like, can we explain like the variability that we see in the genome if we compare to this ancient genome, like how does it vary, and is it like well explained with this population or not? And so that is not a part of my study. I still compared it, and I use the fact that we have a lot of modern modern data. So it was easy to say, okay, it's more closely related to this site than another one, because we compare it to nearly 3,000 genomes compared to some other studies that compared to 10. So they were not able to differentiate that. But yes, so the idea that was more developed in other studies than ours was they try to date the different movement. And so they found that the Aryan farmer was about like 5,000 years before Christ, or like 3,000 years. So it was like one of the first movement. It's always complicated to talk about movement because like we know that we found some archaeological site with samples that look like the ancestry in India, and we have the modern day India, but technically we don't have a migration flow, you know. It's just like we find something that is similar when we talked about DNA. But yeah, so there were like sites that were a long time ago at some place that have DNAs that look like modern Indian. So at some point there must have been contact and movement, but it's a bit difficult to know in which way.
So,
and for the step pastoralists, it's more like 3,000 years ago.
Just to clarify, so this population you mentioned, there's 5,000 from now, or like there is a period like 5,000 from now or 3,000 from now?
So it's more like 7,000 from now to 5,000 for now. So
I see.
those it's also those calculations, they also use a lot of modeling, so it would be like the mutation rate or thing like that. So I'm sure, and if we find different archaeological sites, it can vary still. So we have like large confidence in
that's the Iranian farmers that you mentioned, right? And that is the there was so just to clarify, so there was an ancient population before that already. So that's more or less established. Like there is a consensus that there was a ancient population which came to the subcontinent probably, I don't know, like 50,000 or something around that that time, right? And after that, this is the first one. So sorry, I mean, I'll let you just
no, so there were some South Asian hunter-gatherers that were already there, but we don't have any fossil for that. So so what we know is that we have compared it to some like actual population of the Andamanese island, and the ancestries that we cannot explain with either the Iran or the steppe related ancestry are well explained when we compare it to the Andamanese island hunter-gatherers. So we kind of know that there were already people there that are related to the Ongi population.
I see.
One question though. So I think one of the popular studies in this domain, which I have read, and there's also a famous popular book on it by Tony Joseph called Early Indians. It's by a Harvard geneticist called David Reich. Have you read his work? Yep. So how different are your findings from what he had proposed then? As in, are they different findings per se, or is the manner in which you have arrived at those findings different? If you could probably explain.
Yeah, so just for a bit of context, so Priya Morjani, which is like my advisor, she did her PhD with David Reich.
Okay.
So
the same clan. So it's like your academic grandfather, I guess.
Kind of. And like we collaborate. Like one of the author of the paper, Nick Patterson, was also in David Reich's paper. So like
he's from mathematics background, right? He's a mathematician, right?
He has a crazy background. Like he was a crypto during the Cold War for Britain.
Oh, interesting. Okay. Well, my area is cryptography, but I guess he's probably remember his name a little bit anyway.
And then he worked for a hedge fund. Yeah, so it's kind of like decrypting DNA now. So we really build up on their work, and so. In our work, we don't have any new ancient DNA samples, so we are really using what they published and the big data sets that they have. And in fact, the model with the three ancestries is also in one of their papers. So in fact, what is new in our paper is that we have more modern data. So we are able to kind of pinpoint where does the Aryan farmer related ancestry came from. But we don't, like, it's not a new model that we propose. We are just like trying to make it more precise.
I see. Precise in what sense? Like, is it the exact division or the timing or?
So like the new findings that we find is that we locate kind of the source of the Iranian farming ancestry to a Sarazam archaeological site. And what we also do is that, as I said, like we have the three ancestry, like from north to south, but we have also other model for like east and northeast of India, and we manage like to fit this Sarasam ancestry to this other model. So it really shows that the Iran farmer movement happened before the steppe movement. So that was kind
So
of
So-
like when you
effectively,
study.
the two ancient hominins that we derive, I think maybe 2% of our DNA from are the Denisovans and the Neanderthals, right? Just, I think for our audience, if you could probably explain in layman's terms some of the models that you use, maybe not the model per se, because that will be somewhat technical, but what are the design principles underlying the models that you use to arrive at this conclusion, maybe to start with?
For the archaic hominid, for
Yes, so
Neanderthal and Denisovan.
exactly. So basically, the broad finding that I lay out for our audiences in your paper, you state that roughly 2% of our genome can be traced back to our ancient hominins, which is Neanderthals mainly and Denisovans minorly. 50%, 20%, that's roughly the ratio, right? So if you could probably help explain how you arrived at this finding, this inference.
So what happened 50,000 years ago is that humans migrate out of Africa, and then when they were populating Eurasia, they mix with Neanderthal and Denisovan. And so how we like find the segment is that how we do that? So in fact, Neanderthal and Denisovan were a population of their own, and they have their own history. So if we compare their DNA to ours, there will be way more difference. And
Right.
so when you have a segment or a DNA from Denisovan, you will have way more mutation because it's like old if you compare to other population. So it's kind of, we do it like that. We compare it to the African population that didn't live, so didn't mix with Neanderthal and Denisovan. And then we try to find segments that have a lot of those old mutation, and we call them archaic. So that was the first way to find it. But then, like quite recently, people have been able to sequence Denisovan and Neanderthal DNA, and then we just compare it. Is like in that segment with a lot of mutation, do you find some that are only found in Neanderthal and Denisovan? And that's how we say, okay, so it's Neanderthal or it's Denisovan.
I see.
But yeah.
Yeah, sorry, go ahead.
No, it's just that our study was like people were, in fact, also quite surprised by the amount of Denisovan and Neanderthal segments that we were able to find in India. It was not really studied before. And what was really surprising is that, so the percentage is kind of similar to what is found in European. The percentage of Neanderthal is higher in East Asia, for example, but it's the diversity. So if you like sum up like the different segments that the different people in India have, you have like the highest amount of Neanderthals that was seen before. So it was quite surprising for that. And so we are really wondering why. We know that India is really diverse, so it's also why it's so interesting to study. But so is it because of that, or is it because that maybe the like encounter with Neanderthal was close to India, so that's why there are such a diversity? So it's kind of new questions that we can now ask.
So maybe for Denisovan, there are other countries that maybe have more diversity, is it?
Yeah, it's like it's mostly seen in Oceania, in Papuans.
Papua New Guinea and
Yeah.
Oceania group. Okay, makes sense.
Okay. So it's kind of, when you look at the world map, it kind of makes sense.
Just one question though. This entire study of, and this series of studies of tracing back our roots, right, in some form or shape. I think it stems from genetics, then you move to population studies, migration studies, and eventually everything boils downstream to culture, right? And that's where most of your popular mainstream debates happen. This is something that happened, or rather came to India rather late. So just curious, is there anything specific about Indian conditions that make this data more scarce? Is it the climatic conditions that make the data more scarce? Is there more genome data, ancient genome data in other countries or regions?
So yeah, for the ancient DNA, it's better when it's cold and not humid. So yeah, that's yeah, we don't have a lot of ancient DNA from India, but I know that it's something people are really interested in. So we hope we will try to get to that. What is really nice with that, yeah, the population and the culture is that, for example, for the Sarazam finding about the Iranian ancestry, we found it only with like looking at genetics, and then we contacted some archaeological people, and so like, yeah, of course, like that totally makes sense. And like, oh, okay, so
Okay.
so in fact that's really fit well together. But yeah, I think it's. Yeah, I think ancient DNA is also still quite new, and so we still there are still like a lot of method development that is made to try to get as much DNA as possible from different samples. So I think we may be able, but for now it's a bit of the limitation for the studies.
Yeah, when you say that you look for large database, this large database is corresponding to the modern DNAs, not the ancient one, right? The ancient is anyway scarce.
Yeah, so the ancient is scarce by the number of sample from different places, but also by the DNA quality. So for modern data, we can have like the 22 chromosome and the sexual chromosome of like every individual in quite good quality. Like we are quite confident of like every mutation we see. Because for example, every mutation, every position in the genome is like seen like second 30 times. And in ancient DNA, if it's one time, it's already good. So it's kind of like you have to, yeah, you really have to do either like some like, yeah, chemistry thing when you sequence, but also bioinformatic filtering just to be able to get the DNA.
And is there any sort of law of land? I mean, in David's book, I think I read it a few years back. I think that book's name was like "From Where We Come" or
Who
"Who
we
We
are and
Are."
where we come from.
Yeah.
Who we are.
It's very nice reading actually. It's slightly technical in the beginning, but I think he mentioned a lot of frustrations with like the acquiring of ancient DNA, collecting, and even if like you already mentioned their scars, and because of like conditions like humid conditions makes them even worse, the quality wise, right? On top of that, there are issues. For example, there are laws that kind of prevent acquiring DNAs. Like for example, I think in Israel, I think DNA testing is somehow not permitted at all. I mean, so all this kind of. So, did you encounter any of this in your research, or you have any experience with this?
I think in our study it was not the case at all because it's modern. So modern, you need a lot of authorization. So like the sampling process was really a long process, but at least you get directly like when you have the consent of the person, you can do it. The only thing is that he had to be sequenced in India. It was really nice way to involve people there. But yeah, and in fact, in that study, we are not the one collecting. So our study is part of a really larger study that is like the larger study is named LASI. It's for like longitudinal aging study in India. So they are looking at not only DNA, like economic, like a lot of different social status for like how people age, and they are looking in India. They have like a sample of 73,000 people, and then in that there is another study called LACIDAD, which is mostly focusing on dementia. And then they were looking for the DNA involved, like the genetic factor involved in that. So that's where we arrived. So they sequence DNA and to look at genetic factors and understand like how we associate to phenotype, you have to understand where does it come from, because one mutation in one European or one Asian will maybe not have the same impact because of what's happening somewhere else. So that's why we wanted to really understand what the DNA look like. But for the like difficulties that is encountered, So I know that, for example, they had some sites in Russia that they are not accessible right now. So it's also a lot of politics around all of that. But for like Israel, for example, I know that they are starting to manage to get a DNA from sediment. So not from human, but for sediment. That is also maybe more available, but there will be some laws around it because you still get the DNA of a person, which is a lot of information. But yeah, it's a lot of like technical advances too that help getting more DNA.
Double clicking on that bit where you analyze the ancestry of archaic hominins, right? In current, in modern day genome sequences. You do mention that there are some subsequences called deserts of sorts where you do not have a direct heritage per se from ancient hominins or archaic hominins, however you'd like to define them. One of them, which I found very curious, was FOXP2, right? Which you mentioned is responsible for faculty of language and related attributes or traits in populations. This directly harks back to the debate that's very common in India, right? So in India, just to give you a brief glimpse of the debate, people say that although you had migrants coming to India, and you cannot deny that there were successive waves of migration to India, the hypothesis of the Indian far right is that the original Indian inhabitants were responsible for the Vedic language, and they were a fairly advanced population who did not actually imbibe a lot of cultural attributes from the so-called invaders or foreigners who migrated to India. So I'm just curious, isn't there a direct linkage between this desert, FOXP2, the language-related attributes stemming from that, and this hypothesis? Or
it's it's also called out of India theory. I think that's kind of a common term. Yeah.
So that I don't really know, because I think the FOXP2 is not seen in human. No, it's seen in human, but not in from Neanderthal and Denisovan in other population as well. I think one of the things that we have limitation with when we look at the DNA in people today is that the DNA had to survive. So maybe there were people before, like surely there were people before, but if their DNA is not transmitted to people nowadays, we cannot see it.
That's a good point. Yeah.
And so, in fact, even when we look at the out of Africa migration, so we were looking at, was it like one wave out of Africa to India or a second one? There's like, if it's a really like small wave, like not small wave, but if the DNA that is present is like under 0.1%, it's possible that they come from like kind of anywhere and we don't know it. But so it's always the limit. Like what we can know about is that, but like DNA that passed. So maybe there were people that like we just don't see trace of it, but maybe they mixed too and they just was diluted by other mixture and we don't see it as well.
Yeah. In fact, in David's book, actually, if I now I recall as we speak, that there was a very interesting statement that the steppe population that's coming from the Central Asia between Black Sea and the Caspian, right? And that sort of also replaced a lot of existing DNAs or existing population, even in Europe, like in the Central Europe, Western Europe, and the same population probably like a good, like a fair bit of like I mean, it's not sure what's the number, like ratio, but like a strong one, like it is strong steppe population that migrated, and their DNA. I think if I remember correctly, I think the it was called R1A1 genome, or maybe I'm misspeaking, but that's kind of prevalent also in like more in North India, and sort of it declines in South India. So I guess if you have something to say about it.
So I don't remember exactly about that, but it's quite funny how like it's like, yeah, in Europe and in India, it's kind of the same like distribution. It's like some Iran farmers that migrate, and some steppe pastoralists, and like local hunter-gatherers. So in Europe, it's like European hunter-gatherers, and in India, South Asian hunter-gatherers. But there are also some like things that passed a bit everywhere. So it's not part of this article, but we are also working on the lactose persistence in India. So the ability to digest lactose, that is really prevalent in like North Europe and a bit less in South Europe. And it's also more present in north of India and is less present in South India. And like the first time this mutation was seen in an ancient genome was it like more like Central Europe 8,000 years ago, and that we have like a lot of idea about how it gets selected, because like people that live with like have domesticated animals that them may drink milk a lot from it, and it can help. And so you can really see that even in India there is like that mutation that was, maybe it can happen a second time, but it was first seen 8,000 years ago in Central Europe, and and it's really prevalent in Europe and a bit less in South Asia.
And just for our viewers who basically will jump on this saying people in the south have curd rice all the time, curd is lactase and not lactose. So maybe people who are lactose intolerant,
they can have curd, right? Yeah.
In fact, we
interesting.
were quite surprised, by that, by the like when we look at that mutation, like technically people from the south are not able to drink that much milk, but then we have also like a diet questionnaire and say like, but they drink milk every day.
Yeah,
So
like in Bengal, for example, it's kind of funny. Like you get this, the tea we drink. As you speak, I'm drinking tea right now, and we always, I mean, most of the time we drink it without milk. Although, like there are like both variants available. But if you go like north and west, I think you have mostly one variant of tea. I mean, that's like my experience. Like Srinjoy lives in North India right now. I mean, it's mostly milk tea, right? And they have like a lot of milk and other kind of things available all the time.
Yeah, so exactly as Pratyay was saying, in the north, it's almost akin to blasphemy for people to have tea without milk, while for most of us, having tea is a very solitary exercise. We don't like mixing it with milk per se, at least not me, definitely.
I mean, it depends, but there's a like a large trend of not having tea with. I mean, also like in these street tea shops, right? There you get like both variants typically. And I was just visiting like central India, Indore, and there I was trying to get a black tea, and it's like not available in any of the places actually. But it's kind of nice. I mean, one of the thing is like how this genetic study actually, it's also very useful in tracking several disease. For example, prostate cancer is one of them, right? Probably diabetes, like South Asian population is more prone to diabetes and so on. So I'm not sure if you have actively worked on it, or if you have some study on this that you can talk about in with respect to Indian population.
So I have not actively worked on it, but it's true that when one of the really interesting thing about India and how diverse it is that we are able to see like variant or mutations that was not seen before. So it really like help also to understand like how those genes work, or like, because if you have a mutation that was never seen before, like how does it affect it? What is the phenotype of the people? So it's it's a really rich source. So I know that some study, like there is a big study that is Genome Asia, that also I've looked a bit at that. It's after you also now need to like test, it's kind of candidate for like we call it loss of function variant, and to try to know a bit more. There are still some interesting things that can only be seen in India. So we had like one mutation that was affecting like a risk of like you will need, if you have this mutation, to use another drug to anesthesia, because you can have like bad reaction to it, and it's like not that present anywhere and except in the Vaishya people. So it's like kind of important to know like what is the genetic basis of that, and maybe you can like be more like, I don't know, maybe monitor better the anesthesia if it's people from the ancestry. It's a bit complicated, of course, but at least we try to know more about it. And it's also kind of limitation of the genome study right now. It's like most of the people that are studied in the big, like we call it G1, so GWAS, genome-wide association study, are Europeans. Like it's, when you look at the different proportion, it's quite quite crazy. And so in fact, most of the things that we learn about like the association between phenotype and genotype, it's about European. So it's quite, in fact, limited, because it's not that easy to apply it to other population, but also because we kind of like not look at nearly the entire world, so we can really learn way more by looking at more population. But so that's also one of the things that is nice about our studies, that by looking like kind of deeply at all like the local ancestry and also the archaic ancestry, we kind of like try to put numbers to say like, okay, that can affect, that cannot affect. And so after that, you can maybe have more like disease tracking or like phenotype, genotype analysis.
Maybe just double clicking on where we hail from and where most of the viewers of this podcast hail from, which is the east of India, right?
Yeah.
So the broad thesis is ancestral South Indian, ancestral North Indians are West Eurasian in origin in some form or shape, while we have more similarities with East Asians. So what's the broad proposal here? We have more similarity with, say, the Chinese, the Vietnamese. Who are the people we are similar to?
So in fact, in East, we find like different ancestry. Some were related to more like the people that speak Austro-Asiatic language. So they were a part of that. And then for the East Asian, I haven't looked at like the ancient DNA, because there are less ancient DNA present in that part, like that is available for now. So I cannot really like pinpoint on a population in particular. It's just that it was better explained with the East Asian. I think we use Han Chinese for that, but it was not like, it was just to have a global idea. It was not really to pinpoint on the population.
Yeah. Also, I think that like for the Bengali population, I mean, I think the idea is like a more ancestry compared to other parts of India from this population. It's not that fundamental constituents are different. The fundamental constituents are still some mix of this ancient North Indian and South Indian. On that, actually, I mean, the next question, I think I also, I mean, I read about in some article that there is this caste line, you know, in caste system in India, and there are a lot of this caste system started probably maybe like 2000 to 3000 years back or something along this line, maybe. And there are also huge genetic differences. That's I remember one thing from David Reich's book that was saying that the difference within the same village among different castes in India, genetic differences are more than, for example, I don't know, like a Portuguese and let's say someone Scandinavian, right? So which is very surprising to me. So if you can elaborate on that.
So in our study, we don't have the exact caste label. We just have like the like what the government will choose to decide. So it's like it's a scheduled caste or a scheduled tribe. But that's what. So in fact, that was quite different from like other study. Like for example, the Genome Asia one that really has label, or the previous study from David Schweiz's lab with that really as a caste level. So we were not able to explore that, but it's true that it's kind of like how we define population is like people that mix between each other. And so if people don't mix,
yeah,
even if they live in the same place, they will not share DNA.
Mm.
So it's kind of retracing, which is quite crazy, even more when you like. I think it's a big study in the UK that were able to know like the zip code from someone's DNA, and yeah, it will never be possible to do something like that in India, which also make it like so diverse and so interesting.
Is the caste linked with maybe the percentage of Central Asian steppe pastoralists? Is that something
Like
that
upper
defines caste?
caste more.
Upper caste has more of Central Asian steppe, while lower caste might have a lower
more.
contribution of
Yeah.
that particular set.
Yeah,
Okay.
yeah. So we see a correlation between the caste group and the percentage of the different ancestry. So that's also why that we think that the like kind of those migration may be linked with like the start of the caste system. Because if it was before, there should not be any difference. So that's kind of help us date thing too. Like, okay, so what happened before or after, if we see a correlation or not.
Yeah.
So that's why we think that this mixture is really linked to also the spread of languages and of the caste system in India.
The other interesting thing is that like this division mostly comes with the male DNA, right? The Y chromosome. Like if you trace only the female source, like only the X chromosome source, I mean, typically you do not have so much diversity, right? Is that true or?
So in our study, because we don't have this exact label, we don't see any difference between male and female. But it's like, as I say, we don't have really. It's really what like we have like 4 categories of caste, so it's not something we can really look carefully. And we don't have the Y chromosome. We didn't look at the Y chromosome yet.
Oh, it's only based on the X chromosomes that you have.
Like no, the
Like you
22
do not.
autosomes.
Oh, all this I see. Okay. I'm
Yeah.
not it. It's maybe more technical than what I know, but so that's I think like part of it is also like there was this some article again, like a lot of these things, there are technical parts and then there was like journalistic articles, and there are of course some gaps where like as a non-expert we sometimes fumble a bit. But I remember there's some article about I think it was Crawl or something, like talking about that it was more, I mean, this steppe population was more, it was a dominant population, like more successful in sort of finding their partner in the local female than the local population, or something like this.
As in
But
vis-a-vis the South Asian, like indigenous hunter-gatherers per se, which in this case probably could have been the Harappan or the Mohenjo-daro civilizations, maybe, not very sure.
Indus Valley civilization, yeah.
Roughly, yeah. Those cities, Indus Valley civilization, the Bronze Age. So yeah, that's broadly the thesis, and most of the culture was in India from that period, just stem around this one leitmotif point.
Yeah. It's also interesting is that like this after this paper on ancient DNA was published, I think it was a one science paper by David Reich's group. So Nick Patterson was there, I think, and then one
And
paper.
Priya was
also there,
like,
probably. Okay, that's like okay, and then there is one paper in Cell, and like after these 2 paper published, I mean, there are like conflict between co-authors about some like the conclusions about the thing, and it was very like interesting.
And it was the cultural conclusions. Just to note, it's not that people are doubting the hard statistics. It's what you can infer culturally from the statistics, which is a contentious topic due to obvious reasons.
Yeah, it's like, yeah, I prefer to focus on what the DNA can tell me, what the DNA can tell me. But yeah, it's always a bit complicated because we see correlation, but can we really, infer, like when we just talk about movement, like can we really infer away, like, it's
Right.
always a bit complicated.
So similar things are there in like other parts, like for example in European DNAs, like do you have this kind of cultural conflict, like fights over the results, or it's more in India?
I think there is always a bit of fights. Sometimes people don't like when we say that they are not 100% from Europe, but
I see. Yeah, I guess we can pretty much understand. Yeah.
Yeah. So I think, yeah, there are still a bitter part. I think the things that help a bit in Europe is that we have a lot of archaeological sites. So at some point, when you have a lot of data, it gets just easier. I think we'll see what will be said in like 30 years about Indian DNA, but if we find a lot of archaeological sites and maybe like can have a better idea of like the time scale, because it's always like that if we find like recent archaeological sites with a mixture that's different or that corroborate with one of the theory, it's always gets easier.
Right. Right. So the one thing I want to ask that like when you like write a technical paper, like you probably give some margin of error, right? So say that this is the conclusion with these margins of error. So in your study, what is the margin of error, and is it like because you're studying the modern DNAs? It's my understanding you'd have less margin of error compared to the ancient DNA studies.
I think it depends on what we are looking at, because if we are comparing it to ancient DNA, it really depends on the number of samples. Something that is also different is that, so for our study, the idea was to have really a representative of like whole India, of like, yeah, different people from different region, from like urban and rural area. And it's quite different from like previous studies that were like maybe focusing on some group and like sample like 20 individual in those groups. And this one is more like, yeah, as I said, like a kind of socioeconomic and genetic study. So that's something that is different, and how you sample can also affect like your findings. So for example, for now, we have like 75 individuals for the northeast of India. So that's also why we may not be able to have like huge conclusion about it.
Yeah.
And yeah, and we are like, I think one of the first study to like even sample those population. So it's always like that, like the more you sample, the more diverse you have and individual. So it will help to have a better idea of what is exactly happening. And as I said, we didn't look for now at the Y chromosome. And a huge limitation too is like when we compare to ancient DNA, we need ancient DNA data.
Compare, yeah. And yeah, when there is a sort of news article that typically non-experts like us read, like and you read, like do you find, like from my area, I find like when a lot of the times what happens is that when something is written by a journalist who is like not an expert or something, there is a lot of misleading facts, like there's oversimplification and then there's misleading facts and certain rhetorics are being pushed. So do you see these kind of things in your reports that cover your results?
So most of the time they contact us as well, and they ask us question, and because they know that they are not experts, they like double check with us. So I think at least for our study, it was quite good for that. Yeah, I know that it can be quite difficult because people sometimes want like catchy result and like big phrase, and we're like, but
Yeah,
maybe
exactly.
in 10 years we'll find like
Temper it
other
down.
ancient DNA and everything will be said differently. Like it's kind of like for now with what we have available, either from the DNA side, but also from like model side and statistic sides. That's what we can say, and that's like our best theory for now. But it's always a bit complicated to say that.
Right. So I think we're probably close to wrapping up, but I want to ask you some just out of the box thing that, do you have techniques like machine learning applications that can revolutionize, like it's apparently changing a lot of other fields. So just wanted to check.
Yeah, there are like a lot of machine learning methods that are applied in population genetics, genomics
Oh,
right now.
already, I see. Okay.
Yeah, but it's still in progress. So in fact, one of the thing is the chromosome painting one. So if you will give like two population to the machine learning model and know like, okay, this population is from that ancestry, this population is from that ancestry. Can you find me on this genome like the parts that are from each like population? So it's worked really well, for example, for African American. So they will give like a training reference of like African
I see.
population, a training reference of European population, and they will give the genome of African American and say, okay, can you find the different parts? And it's really a classification. So it's
Problem, yeah.
really like what machine learning is good for.
Right.
So
So
I
I
think
know that
your paper uses PCA, right? You use principal component analysis and HMMIX, which is the main model from which many results stem from. I think it is also a Markov chain based model with probabilistic switches right from chase to chase.
Yeah, for the archaic part, it's really it's a hidden Markov model that try to find that look at the density of the mutation and like is the density similar or is it different? And when you see a big switch, you say, okay, this was modern human and this is archaic. And it's also used to color the chromosome about the ancestry that okay. So this will not be based on density, but this will be like, what is the probability that I copy from that reference and does it switch? So yeah, it's kind of what I said, that there are a lot of nice like probabilistic and CS problems in genetics, in genomics.
So progress in machine learning is likely to like help your kind of research as well directly.
Yeah, yes, for sure. After I think one of the things that is maybe a bit more difficult is like when it's just classifying, I think it will really help, but it's just sometimes we need to understand the process that in behind and that like it's
That's
still
hard,
possible with
yeah.
machine learning, but you have maybe to give the right summary statistic or thing like that. So I think sometimes people think it's a bit too magic, but I think it's always like that with machine learning, but at least for the classification one is like it's kind of what is it made for? So it will really help.
Right.
But for example, we were not really able to use that for India because, as I say about like the three main ancestry, and for the step one and for the Iran farmer one, we have like DNA, ancient DNA. But for the South Asian, like hunter-gatherer one, there are no
like the Andamanis, you do not have a lot of DNA.
The Andamanis we have, but they're really like they were on an island for a long time, so they're really drifted from like we know that
lots of mutations, right?
Yeah. So if it's for classification, it's not good. We still like can find with like other statistics that there are similarities, but we cannot use for like local classification. But that can change like if we have like this can be solved either by the DNA sample part or by the like more method part. So that's kind of like two way to look at the problem.
All right. So I think we can just leave it there. Here, I mean, Srinjoy, if you want to say something,
Just
ask.
one question, very orthogonal question though. So uh, for someone like me who's probably interested in mathematics, was scared of biology all his life, because in India we really
You
do
gotta
not have
choose.
you have to choose, and you don't have an education system that lets you appreciate
Not yet,
early
yeah,
on
but yeah.
the interlinkages between different disciplines, right? Is there some dummies guide to genetics that someone could pick up, some books that you could probably refer to, where an interested reader could learn more about your field, probably learn about the problems that you guys are tackling now, stuff like that?
I don't know. Yeah, I think there must be some textbook about like statistical genetics that can be a way to get into. But human evolution, I know that Jonathan Pritchard from Stanford has like it's also online and free. He released like half of his textbook, so that can be a nice start. But I think that I understand that biology can be quite scary, but I think biology can also be more like an horizontal field. I think for me, it's not like mathematical when you like really need to have all the bases to go up. I think it's also a bit the case in genetics, of course, but I will think that you have to not be scared, because in fact most of the biologists are scared by the computational parts. So in fact in Berkeley there is a graduate program like for PhDs that is named computational biology, and I think it's people from either like computing background or biology background or more statistical background that can apply. So I think computational bio people know that it can be really valuable to have someone that know maybe more about computing and less about biology.
So, I mean, as a discipline, like, it's an emerging field, like this combinatorial mathematical applications to evolution and all. Like, I guess, like there is a, of course, this ancient DNA study and like population studies, one applications. The other thing can be like studying, like tracking diseases, for example. I think so. My interpretation is that it seems to be like an emerging field. Like, a lot of people can also get into that, right? So just to understand your feeling about it.
Yeah, I think it's getting like bigger and bigger with like quite different things. Like, on-chain DNA brings new problem, like what if you have low confidence in your genomes? Like maybe you need some likelihood model to manage to infer things. But we also have more and more samples. Like, for example, the big UK biobank, it's, I think, at least more than 400,000 genomes. So
Wow.
I think we can, like, if you know a bit about computing, you know that it's not the same problem to compare them than when you have 1,000 or so. It's kind of all those things, like it's getting bigger in terms of sample, but also more diverse in terms of like quality of things. And like also if you have something that is ancient, how do you compare it from something that is modern? Like it's a lot of like interesting, more like methods question. And and it's just like about structure of population. But you have also, like the disease tracking, like it's a lot of statistics too. Like how do you see correlation between mutation or combination of mutation and phenotypes? It's also a lot of statistical development for that.
All right, then. Thanks a lot, Elise, for giving your time. It's a Sunday at your place, I guess, Sunday morning. So thanks a lot for finding times for us. And it's been very helpful. It's been very nice explaining all these things. I really wanted to talk to some, someone working in this area for long, and we fortunately found you. So great talking to you. And since I was also a postdoctoral researcher at Berkeley, so I can also see you as one of my, junior that's working in the same thing. I guess you're also part of this UC postdoc union. That's very good, right?
Yeah.
They're very helpful. Yeah. Okay, great. Really good luck in your career, and hope to see more of exciting results in this field, and we'll be waiting for that. Thanks a lot. We just leave it there. Okay, bye-bye.
Thank you. Bye-bye.
Thanks.